In Chapter 1, Part 4, we are going to start off talking about what bacteria like to eat. Bacteria all have three major nutritional needs for growth. That is a source of carbon for its cellular constituents, a source of nitrogen for proteins, and a source of energy, or ATP, for cell functions. Microbial growth and nutrition are divided into two groups, autotrophs and heterotrophs. Autotrophs can grow using carbon dioxide as the only carbon source, and heterotrophs require an organic source of carbon, like glucose, and get energy from fermenting or oxidizing organic substances. Heterotrophs can use glucose as the carbon source and the energy source. All bacteria that inhabit the human body are heterotrophs. Now the fun, colorful part. There are many different types of growth media used in the clinical microlab, and these media are they start with an agar base. So when we say agar or media, we are talking about the same thing. And so media is the plural form of medium. So one plate would be medium, and a whole bunch of plates would be media. So there are many different types. So don't get confused or overwhelmed. We're going to cover this quite a bit. But just look at all the different types of culture media. There are all these different types of agar or media. Each one of these has a different nutrients or carbon source or sugar source, lactose or glucose or fructose, or that's what they have in them, a different source of carbohydrates. And in these different slants, you can have agar slants, or you can have liquid broth media. So all of these represent something different. And then down here on the bottom picture, you can see different bacteria growing on the different media, and they grow in all different colors depending on what medium they are plated on, and it's so interesting. So minimal medium is very simple, and it's not usually used in the diagnostic lab. Nutrient media contains meat or soybean extract. Then we have enriched medium, selective, differential, and then transport medium. So we're going to come back to this one. Nutrient media, as I said, contains meat or soybean extract. And an example of a nutrient media is nutrient broth such as trypticase soy broth, and also trypticase soy agar. So this would be the broth in the front, and then this is the agar in the back, these plates. Now, and this is a great picture because you can see that there is a bottom and a lid. So the agar is in the bottom, and the top is covering. So what we do when we incubate these, we flip these upside down and we label the bottom where the agar is. So that way if the lid flies off or we drop them and we lose the lids, at least we've still got a label on the bottom. That's important. Okay, so the next type of media is enriched media. And this is, you take the basic recipe, the basic nutrient media recipe usually, and you add some type of factor, some type of growth factor like blood, vitamins, or yeast extract. And examples of the enriched media have blood and chocolate. So blood agar contains 5% sheep blood. So these are beautiful pit cultures. So I'm going to tell you what these are. These are blood agar plates, probably growing gram-positive cocci. We would only know that if we did a gram stain or a quick biochemical test. But you see that there's a clearing behind the colony. See the pinpoint, or the little white dot, is the colony growing in the middle, and the area cleared around is where the bacteria has hemolyzed or destroyed the red blood cells underneath, and you can tell that these colonies are white. On this plate on the right, See how it has a yellowish tint? It's also beta hemolytic, but this is probably a Staphylococcus or Micrococcus because those have a yellow or golden color. So this is the kind of stuff we're going to look very closely at our bacteria, how they grow, what they look like. It's just so interesting. Okay, also this is another picture of enriched media, chocolate. Chocolate agar is made from this sheep blood agar, but it's heated gently to lyse thread cells, releasing X and V factor. Those are also called Hemin and NAD, and we're going to get into that later on. You don't have to write that down now, but just know that chocolate agar and sheep blood agar are enriched media because they are enriched with blood. And we're going to learn later on that in chocolate agar also has some antibiotics in it. Now, selective media contain additives that inhibit the growth of some bacteria while they allow others to grow. For example, MacConkey agar, which will support the growth of gram-negative bacteria, inhibits the growth of gram-positive. So this bacteria up here that I said is most likely a gram-positive, it's not going to grow on MacConkey because MacConkey is selective for gram-negative bacteria. And it contains crystal violet, which inhibits gram-positive organisms, and it also gives it this beautiful purple color. So we say that MacConkey is selective for gram-negative bacteria. This is another example on the right of a bacteria or an agar that is selective for gram-positive. This is called colistin nalidixic acid, or CNA, and it is a blood agar plate, the same 5% sheep blood agar right here, and they've added these antibiotics, which select for gram-positive. So these colistin nalidixic acid will kill gram-negative bacteria most of the time, allowing gram-positive bacteria to grow. So the antibiotics it contains is colistin and nalidixic acid. And if you can learn these little tidbits now while we're going through chapter 1, that'll be one less thing you have to learn later on, because that will be something definitely that you do need to learn. The next type of media is differential media, and this contains ingredients that allow us to visualize metabolic differences. Remember, I told you way back in the beginning of chapter 1 that we have to know bacterial structure and physiology. Well, the metabolic process is part of the physiology. So this MacConkey agar, remember, it's also selective, but it can be selective and differential. So it is differential because MacConkey allows us to determine if a bacteria is a lactose fermenter or a non-lactose fermenter. That means, can it use lactose? Can the bacteria use lactose as a form of carbohydrate or energy? So the lactose fermenters turn pink on the agar. Now, if you have a non-lactose fermenter that cannot use the lactose as its carbon source, you will have a clear colony. So pink for lactose fermenters, clear for non-lactose fermenters. And this is a huge step in identifying gram-negative bacteria. As a matter of fact, it is the first thing that we were going to look at when we start doing our bacterial cultures for gram-negative bacteria. That's going to be the first question I ask. Does it grow on MacConkey? Yes. And if it's a gram-negative rod, that means it's growing on MacConkey. And then my next question is going to be, is it a lactose fermenter or non-lactose fermenter? So this, again, is a very, very important point. Now, we're going to go back up to and talk about to slide 32 and talk about transport medium. Transport medium is a holding medium that will preserve bacteria but not allow it to grow. An example of transport media are Stewart broth, Amy's broth, and Carrie Blair. So those are the 3 types of transport medium, and we will be seeing more transport medium and hearing more about those later on in the course. So we're about at 10 minutes, so I'm going to end part 4 right here.